Hi students, I am Shaileyee welcoming you to the Medical and Pharmacy Crash Course division in the Knowledge Club online. Students, today I have come with an episode in the Medical and Pharmacy Crash Course division, the quick revision of cell structure and functions chapter. These question answer sessions will help you to ready for any kind of competitive exam or medical exam. I divided the quick revision of cell structure and functions chapter in 3 parts for making the short episodes. This is part 2 episode. I discussed 40 suggestive terms in the previous episode. These terms are very important for your exams. Today I have come with another 40 important terms in the cell structure and function chapter. Maximum terms are decorated in alphabetically for easy to memorize the terms for you. These episodes help you to revise, consolidate and polish your knowledge and skills. Also help you to develop an in-depth understanding of the subject, assess and review knowledge. Suggestions contained in these types of episodes have been designed to be similar in the pattern used in JEE Medical, NEET, CSIR NET, GRE, DBT, JRF, ICMR, GATE, GPAT, JNU PhD, JU PhD, IAS and so on, medical exams and pharmacy semester exams, and obviously in the WBCS and IAS Prelims exam. For the sake of better self-assessment as well as a thorough and comprehensive understanding of the subject, I have kept it topic or chapter wise. Stay tuned up to end. If you skip, you can miss a valuable points for your exam. So stay tuned up to end. I am going to start today's episode. Let's start. So today's first point, the Golgi apparatus. The cellular organelle formed by a series of flattened membrane-bound sacs that functions in protein modification, tagging, packaging and transport. Point number 2, the gene expression. What is gene expression? The active interpretation of the information coded in a gene to produce a functional gene product. Point number 3, gene. Functional length of DNA that provides the genetic information necessary to build a protein. Point number 4, genome. The entire complement of an organism's DNA found within virtually every cell. Point number 5, glycocalyx. Coating of sugar molecules that surrounds the cell membrane. It's called the glycocalyx. Point number 6, glycoprotein. Protein that has one or more carbohydrates attached. Point number 7, the helicase. Enzymes that functions to separate the 2 DNA strands of a double helix during DNA replication. Point number 8, histone. Family of proteins that associated with DNA in the nucleus to form chromatin. Point number 9, homologous. Describes 2 copies of the same chromosome, not identical. One inherited from each parent. Point number 10, hydrophilic. Describes a substance or structure attracted to water. Point number 11, the hydrophobic. Describes a substance or structure repelled by water. Point number 12, hypertonic. Describes a solution concentration that is higher than a reference concentration. Point number 13, hypotonic. Describes a solution concentration that is lower than a reference concentration. Point number 14, integral protein. Membrane associated protein that spans the entire width of the lipid bilayer. Point number 15, the intermediate filament. The type of cytoskeletal filament made of keratin, characterized by an intermediate thickness and playing a role in resisting cellular tension. 16. Interphase. Entire life cycle of a cell excluding the mitosis. Point number 17. Interstitial fluid, the IF. Fluid in the small spaces between cells not contained within blood vessels. 18. Intracellular fluid, ICF. The fluid in the cytosol of cells. 19. Intron. Non-coding regions of a pre-mRNA transcript that may be removed during splicing. Point number 20. The isotonic solution. The isotonic that describes a solution concentration that is the same as a reference concentration. 21. Kinetochore. The region of a centromere where microtubules attach to a pair of sister chromatids. Point number 22. The ligand. Molecule that binds with specificity to a specific receptor molecule. The ligand-gated receptor, ligand-gated channel. It's called the ligand, the molecule that binds with specificity of a receptor molecule. 23. The lysosome. The membrane-bound cellular organelle originating from the Golgi apparatus and containing digestive enzymes. 24. The messenger RNA. It's also called the mRNA. The nucleotide molecule that serves as an intermediate in the genetic code between DNA and protein. 25. The metaphase plate. The linear alignment of sister chromatids in the center of the cell which takes place during metaphase, and which called the metaphase, the second stage of mitosis and meiosis, characterized by the linear alignment of sister chromatids in the center of the cell. Point number 27. Microfilament. The thinnest of the cytoskeletal filament, composed of actin subunits that function in muscle contraction and cellular structural support. Point number 28. Microtubule. The thickest of the cytoskeletal filaments, composed of tubulin subunits that function in cellular movement and structural support. Point number 29. Mitochondrion. One of the cellular organelles bound by a double lipid bilayer that function primarily in the production of cellular energy, that is called ATP. Point number 30. Mitosis. The division of genetic material during which the cell nucleus breaks down and two new fully functional nuclei are formed. Point number 31. The mitotic phase. Phase of the cell cycle in which a cell undergoes mitosis. Point number 32. Mitotic spindle. Network of microtubules originating from centrioles that arranges and pulls apart chromosomes during mitosis. What is called multipotent? That describes the condition of being able to differentiate into different types of cells within a given cell lineage or small number of lineages, such as a red blood cell or white blood cell. 34. The mutation. Change in the nucleotide sequence in a gene within a cell's DNA. What is called the nuclear envelope? The membrane that surrounds the nucleus, consisting of a double lipid bilayer. Nuclear pore. One of the small protein-lined opening found scattered throughout the nuclear envelope. 37. Nucleolus. Small region of the nucleus that functions in ribosome synthesis. 38. Nucleosome. unit of chromatin consisting of a DNA strand wrapped around histone proteins. Point number 39. Nucleus. Cell's central organelle contains the cell's DNA. And the last point, the 40. Oligopotent. Describes the condition of being more specialized than multipotency, the condition of being able to differentiate into one of a few possible cell types. This point is the last point of today's episode. So guys, hope you enjoyed today's important term session and increased your knowledge more on cell structure and functions. I am coming with another revision session on chapter Part 3 in the next episode, which will also help you to flourish your knowledge more. If you like this video, please like, share and comments freely. Don't forget to subscribe our channel. If you already subscribed, then thanks a lot. After subscribing our channel, please click on the bell button for getting the regular updates. I am coming with another episode very shortly. Till then, goodbye.